The title of my talk is 3D Modeling of Zebrafish Embryonic Tissue. I'm Lisa Manning. I'm at Princeton, and this is work in collaboration with Aggregates, who's also at Princeton University. I was going to talk to you today about surface tension and a model for that in tissues, but there's some really exciting new experimental data that makes that still more of a work in progress. And so I'll be discussing this, which is a related topic today. And so here's a few movies of motion in tissues. And so we've been told before that these embryonic tissues behave like liquids on long time scales. And the first movie is pictured in vivo, and this is nuclei staining. And so you can see that the cells are moving in this. And this is, in vivo, it's in what's called the shield of the embryo, so it's the organizer. And some cells are moving this way, but you can see that there's some motion up that way. I'll show it one more time. And so an overarching question is what drives flows in these embryonic tissues, in these liquid-like tissues. And then the second thing I'll show you is an in vitro experiment where these are aggregates of cells now, and they're moving together and fusing just like drops of liquid, basically. I think it's a really neat picture to show that this, on long time scales, this is like over 4 hours that these tissues behave like fluids. But the interesting thing is if you look at the structure, you ask the question, why are these materials behaving fluid-like on long time scales? This is a confocal image of a 2D slice through an aggregate, and blue is nuclei, and green is the membrane. And you see that the structure really does look like a foam, as we've been told before. It's a close-packed structure, and here's some 3D images. And so also you know right away that these cells are big enough that thermal temperature doesn't cause motion in these cells. And so what gives rise to this, if it were a foam like this, in order to get motion, you'd have to shear it externally. The foam wouldn't just move by itself. And there's not thermal temperature here, so how can we model these tissues? Another point is that it's not exactly a liquid. So this is a plot of the mean square displacement as a function of time window, and this is a slope 1 on a log-log plot. So you can see that at long time scales, it is diffusive, but at shorter time scales, it's subdiffusive. And in general, this is sort of an indication of caging or glassy or frustrated motion at these shorter time windows. Oh, and one other point I'd like to make is that unlike other liquids, there's no cell loss. So inside of here, these cells are behaving like liquid, but they don't ever move outside of the aggregate. And so that's also a difficult thing to model. I mean, we'd like a model to capture. And so the question I'll be talking about today is what minimal mechanical model explains and predicts these flows and the structure. And so importantly, I want to make sure that we're doing a quantitative comparison between this model and experiment as well. So I'll talk about that. And so the model that we developed is a cell interaction model. And so we treat these cells we talk about the cell center of mass, and they interact via a two-body potential, which I'll talk about in a minute. There's also dissipation through frictional damping. And the shape changes in this model are passive, which means that the cells change their shape based on their interactions with their neighbors. And where the active processes come in this model is through the active forcing, and I'll talk about that. And also we'll neglect inertia, so that we have an equation for the motion of each cell, which is basically the sum of a potential, a damping, and an active force. And this, for those of you who are familiar, this idea is sort of very similar to a model for foams by Doug Durian, with obviously changes. And so the interaction potential, I think, is interesting because there's other models like this, but they use something like a Morse potential. And what I'd like to point out is that what you really want is a contact mechanics model for how these cells interact. Because you're talking about macroscopic objects which are coming together and squishing. And so the area over which their adhesion acts is very small, but what's important is how much contact area they have for their adhesive properties. And so for this model, I'll use what's called the DMT contact mechanics model. But in general, it's just, and the repulsion is like elastic spheres. So these cells repel like elastic spheres. And the adhesive energy is proportional to the surface area in contact. And that's what this model does. And then also I'd like to talk about the active force. Because as a physicist, I might think that the way to model the active force would be using stochastic white noise, that inside this packing that each cell basically has some noise term that describes the active forcing. But that's not the right thing to do in this case, because this is an interesting system. There's no extracellular matrix like what we've heard about before, and these cells are swimmers. So they can't move unless they exert a force on another cell. And so that right away tells you two things, is that number one is that you have to obey Newton's third law, so if one cell pulls on the other better experience a force. And in addition, that the direction of the active force is biased, because we know that cells move by basically developing new adhesive contacts and moving in the direction of those contacts, essentially under tension. And what that means is you pull in the direction of your neighbor. You never push off, essentially. And so with that biases this active noise that you feel from your neighbors. And so that's also in this model. And so just to flash this up here, this is what the model looks like. It's basically like a position Langevin equation. And there's 3 non-dimensionalized parameters. And so what you can see is there's parameters that we can measure. And then there's 2 parameters, which is the damping and the magnitude of the active forcing, which you can't really measure, and so we'll have to try to get at that in a different way. So first I'd like to show you that, just to give you an idea of what this model looks like when you run it. And so what you see, first of all, is that you get a nice rounding up of the aggregate, and you don't get any cells exiting the packing. And that's actually hard to do, to get diffusion here in the center and not to get them to move away from the packing, and that's because of this bias in the active forcing. The colors are just to show you that they don't mean anything other than to show you that at the end of this simulation, some of the ones that were in the middle have moved up to the edges.
And why they don't move away?
Because essentially, if you had a stochastic forcing, they would, right? But because if their direction that they have to move is biased in the direction of the contacts they're making with their neighbors. Essentially, that's the idea. Also, you can worry, because some other models like this have crystallization, because you have a potential, and under agitation, the minimum energy state for these spherical objects might be a crystal, but that's not what you see. This is a pair correlation function, and for these spherically symmetric particles, if we were a crystal, you'd see peaks here and here. And actually, in some of the simulations, you see what has been shown to be signatures of jamming behavior as well. But the important point is that these things stay disordered. The other thing is we can sort of do something more quantitative, and we can look at the mean squared displacement as a function of the time window. And this is the simulation. This is experimental data. And you can see that, again, at long times you get diffusive-like behavior. At short times you get subdiffusive behavior. This red is slope 1, and this is slope 1/2, just to give you an idea. But then when you extract the diffusion constant with these simulation parameters, it's a little too small. And so what you can do is actually run a whole bunch of simulations with a lot of different parameters, and then figure out what's the, you can measure what this adhesion energy is, and then you can say, okay, well, I know what the diffusion constant is in my experiment, so that gives me one of the parameters that I need for my model. So just give you an idea that we're really trying to quantitatively match up the simulation with the experimental data. And so this is a cell interaction model for embryonic tissues that matches the experimental observations of diffusion plus subdiffusive behavior, rounding up, no crystallization. But we want this to be, we want to make predictions, because a lot of people have shown simulations, and we show that we fit the data, so this is good. It seems to be a good model, but we'd like to make predictions. And so cell sorting experiments that were alluded to before are sort of the sensitive test of the dynamics and the timescales. And so once we do finish this fitting procedure, then we can go and make predictions about these cell sorting experiments, and that's what's up next.
Aren't the peaks in G of R still consistent with a triangular lattice instead of a square lattice?
They're not in 3D. I mean, it's, no. So do you let me go back? So these are basically, in 3D, these are signatures of tetrahedral coordination, which doesn't tile space. So. Does that answer your question? Yes.
In your initial conditions, do you start the cells moving randomly, or do they move in some given direction?
So the way the simulation starts is I generate I generate a random distribution of cell centers, and then I equilibrate the cell centers without any active forcing. So I sort of let them come to an equilibrium shape, which is basically if I started them out in a cube, then they come into a cube, and then I start the active forcing once sort of the shape without the active forcing is equilibrated. And then the active forcing is just by a stochastic white noise from that equilibrated.
So they don't have an initial velocity. They just move under the forces.
They move under the forces, exactly.
You use this DMT adhesion model. Isn't there hysteresis in there, and how is it taken into account in your simulation?
That's a great question. The DMT model I'm using is the one that basically has, I'm using the force goes to zero at zero overlap. You're exactly right that in a real system there is hysteresis, because in this model, essentially once you get close, you go very quickly to the equilibrium position.
So you eliminate this.
So I eliminate this in this model, but I agree it's very interesting and something to be looked at.
Any more questions? Thank you very much. Next speaker is Thomas Angelini from Harvard University, speaking on multicellular substrate deformation guides guiding collective migration.
Yeah, you still need this? Yes, I do. Thank you. I'll give it back to you in a minute.
Okay. Is this working? No? Not yet? Maybe it's just a button or something. Oh, mute. Is this it's okay? Okay. Hi, I'm Tommy Angelini from the Weights Lab at Harvard, and I think really the theme of this talk is just it's like we've heard about a few times today. Basically, mechanical communication for guiding collective motion, and I think it's along the same kind of lines as what Cindy talked about and what Paul talked about. But I think the mechanism here is something totally different. So we've heard about the importance of tissue cell collective migration. I won't go into it after all morning. I guess what I want to say is that these cells, of course, are self-driven, but their interactions are adhesive. And in my case, probably, well, one of the most important attributes is that they're interacting with this elastic medium. And so the experiment is we have a huge island of cells, and we look at a small region near the middle, far from the edge, and just ask, how do groups of cells inside of here move? Do we see any collective motion? Do they move in small groups, big groups, etc.? And as you pack them more and more together, do these correlated domains grow or shrink or what? And so, if from Cindy's talk, we might think, well, her nice paper suggests that a cell contracting here and a cell contracting here feel each other through this transmission of stress to the substrate, right? But as I think has been discussed, if you glue these two cells together by the cell body, you can get rid of that vector there and balance the forces. And that gives you a way to, say, deform the surface over a longer distance, and forces transmitted from cell to cell. And in fact, Javier Tripot from the Fredberg lab, I think, showed this quite nicely, that the forces transmit across the entire colony, and that, on average, this thing acts like one giant contracting unit. However, which is important in my work, there are fluctuations in there, and the fluctuations, I think, are what guide motion inside of here. Oh, and if I could just make one comment. I think that, we've heard lots of nice models of hydrodynamics and particles colliding. I think this particular case, the most, I think, pertinent adaptation of a good classical model would be an active version of the Frankel-Kontarova model, where your cells, say the springs and balls, are active, you have damping, but also they can somehow remodel the potential they see. So that would be a new version. And in fact, what I think is that they're deforming the surface, creating what is effectively a potential that changes, and then therefore changes how they move. So, okay, just to get to it. This is a movie, just a sample of data that we would see. They're NDCK epithelial cells. It's about an 800 micron field of view. We take 16 of these data sets in succession to get many density points. And so you start off at low densities and go to high densities. And this is how they move together. If you look at how they deform the substrate below, you see little teeny fluctuations. It's like little, like blades of grass blowing in the wind or something. It's I don't know. It doesn't look all that dramatic, right? But as we've learned, you have to relax the cells, or kill them, or remove them from the surface. So if you trypsinize the cells, instead of that little short wavelength high frequency motion, you see larger scale deformations. Again, this is the same 800 micron field of view, so you see like big bands that move in the same direction. So there are large scale deformations that propagate from cell to cell and actually move in the same direction. These deformations are no longer balanced within the cell. They're balanced over many cells.
What was the cells?
I'm sorry?
You said that you removed the cells.
Yeah, we trypsinized them.
What motion?
So this is a comparison of a time point at which you have cells on the surface, and then to the same place where you've removed the cells. And I'm just flashing back and forth between those two. Yeah. So the motion never actually looks like this. This is just revealing what's hidden underneath. Yeah. So if you do PIV on those kinds of, if you compare those two fields of view and do PIV, same 800 micron field of view, you do see indeed large scale collective deformations below the cell layer. So to get a length scale out of this, we just calculated a correlation function, something similar to what we heard about yesterday. And we take a displacement at one point and dot it with a displacement some distance r away. And then you average this over all orientations and over all times throughout the data set. And that gives you a 2-point correlation function that's sensitive to changes in orientation and displacement. And in that case, we see this correlation function decays exponentially. And you can just fit a line to this and get a correlation length. And what we see is, as the experiment goes on and cell density goes up, this correlation length grows. So somehow the correlated domains of deformation are going up. And so just to show you a quick kind of flavor of how the cells move, at low densities, their shape is irregular. They're quite violent. They're moving fast. But then at later times, the motion is much more smooth. You start to see more like streams and less kind of violent behavior, but more streaming, smooth behavior. And eventually, you just see lots of big groups of cells that move together. So you can do the same PIV analysis on that motion, and indeed, again, you see large swirly domains, and we use almost the exact same correlation function, but now on velocity vectors. And this time, instead of decaying exponentially, we see it at each time point we have a negative minimum, which means these are more swirly. And so we take this negative minimum as to be basically, if you have a velocity vector here and you look some distance away, they're going in the opposite direction here, and that defines kind of a domain over which they can move together. And so you can see that correlation length grows as well in time, whether you plot it in microns or in units of cell lengths, say. So how are these patterns related? You can overlay the two PIV movies and stare at it for days, which I did, and I couldn't figure anything out. So, instead, why not just plot, say, the correlation like the deformations versus the swirl size? And so, of course, they both grow in time, so they're going to both grow with one another. And they're of the same order. They're quite close in magnitude. So that's our first clue that the deformations are somehow guiding the motion of the cells. Secondly, if you do the same experiment on glass, you can't see there's no deformations in the glass, but you can still get a swirl pattern on glass. And what we find is that the swirl size actually decreases with density on glass, where it increases with density on polyacrylamide. So the deformability of the substrate is critical to this kind of behavior. And then finally, if you want to kind of get a temporal relationship, we can just do a cross correlation in time and say, well, given a displacement, a substrate displacement at time t, what are the odds of getting a big velocity fluctuation at time t plus tau? And then just average this over all positions. And what we find is that basically the most important thing is that this correlation function across the zero lag time point, the slope is positive, which means that velocity fluctuations are always lagging behind deformation fluctuations. So you have a deformation fluctuation, say at some point, and it's going to be sometime later that the velocity vector catches up with it. So and then you can plot the slope of this as a function of, say, time in the experiment or density, and we see that indeed it's always positive and it decreases. So as the system slows down, velocity fluctuations are slower to catch up to deformation fluctuations. So I think the message of that is that cells are communicating long distance. They're contracting in a large scale manner, imposing a deformation field on the surface that acts as some kind of potential field for them to walk on and organize their motion. Now, how am I doing at time? Oh, excellent. Okay, so this next part of the talk is actually what I most wanted to show everybody, because I thought maybe I could get some help from you guys or some ideas, because it's something I'm still a little puzzled on, even though I really like the analysis. Basically, what are the cells doing on the small scale that give rise to all this? I mean, you can say, okay, there's this big deformation, and they follow it, but why do they follow it? And how do local contractions interact with, say, large-scale velocity fluctuations, or large-scale contractions interact with local velocity fluctuations, and these kinds of. So it's a multi-scale and multi-time, scale problem, and also cell shape is really dynamic. It's not like you have a bunch of balls hitting each other and stuff. So basically, I attacked it with the only tool I can think of when you have to find hidden structure in dynamics, which is basically a scattering method. I calculate the dynamic structure factor of cell motion and bead motion. And so just briefly, for those of you who aren't familiar with this, it's basically the Fourier transform of the density in space and time. Modulus squared of that gives you what's called the dynamic structure factor. And for any generalized hydrodynamics allows us to derive this for basically any system that has the right kind of memory kernel and all this. And point is that if you don't know what your fluid's doing, as long as it's stochastic, you'll usually get a dynamic structure factor that looks like this. And this omega of q, capital omega of q, defines a peak position, but we don't know how it depends on wave vector. And this gamma of q defines a peak width, but we don't know how it depends on wave vector. And so for fluids, we know how these things behave. This goes like omega over q, and this goes like q squared. Or rather, sorry, this goes like c times speed of sound times q, and this goes like q squared. But in our case, we just don't know, so we're going to derive that. And then just one final note: it's common to calculate the current version of this, the current correlation function, and it ends up just being omega squared times q squared times this, and it's the exact same thing. In either case, all we really want are the dispersion relations. The dispersion of peak position and dispersion relation of peak width. I know this doesn't sound like a good plan, but the reason you want this, is because you want to know what on some length scale is there a special, or rather, given a Fourier component, given some length scale, is there some special corresponding time scale, and what's the lifetime of that? And furthermore, are there a whole set of length scales and time scales that lead to propagation of things? And this will start to allow you to kind of tease out how these two densities are interacting. So here's a sample of a dynamic structure factor of cell motion. And then if you take a slice along here, you can fit the line shape with that function on the last curve, and the line shape fits beautifully. And so, like if you do it at one Q value, you get like a peak here, and if you do another Q value, you get like a peak there, and so forth. So you get a peak position and a peak width. Now, so what do these things look like? Well, for the cell layer, the peak position dispersion, so we have omega versus Q, and it disperses like this, and the peak width goes like this. So the only reason I want to show these is to say one thing. This motion is underdamped. The peak position is higher than the peak width. Just barely, but it means that maybe density fluctuations can propagate a little bit. Not very far, but a little bit. And if you want to say get some propagation speed, you take the slope, and that's the group velocity of your fluctuation. The gel, however, is always overdamped. The peak width is always much larger than the peak position. So you can never get a propagating mode in the gel. But however, you still get fluctuations, and you get a frequency, and you get a lifetime from the peak width. And so these can all be useful. So if you do this for all of the data sets, they all look like this. The frequency goes down, etc. This is if you plot the gel dispersion on top of the cell dispersion, I think the first thing you see is that they cross at 2 points. And I think this is important because it says that there is a spatial wavelength in both and a spatial frequency in both that match up. Means you can get strong coupling there and there. And furthermore, I thought, well, maybe these all have similar shapes, so maybe these important length scales will allow us to rescale all the data and collapse it. And in fact, you can rescale all 16 data sets in the gel and on the cells, and you still preserve this kind of crossover and shape. And also, what's potentially interesting is there's an energy gap. It says that this mode needs a little kick to couple to this mode, and it makes me think maybe there's a way to change this energy gap and therefore change the properties of the system. And however, that's just I don't know what's that. Okay, this is almost second to last slide. I figure those length scales and time scales are important. So just to get a visual idea of what's going on, I take my data and I Fourier filter it and get rid of all wavelengths and frequencies except for the crossover. And so in the gel, you see basically what looks like just a couple of fluctuating dots going like this. It's almost like an optical mode or something, or a standing wave where just like no speed, no propagation, and if you watch it long enough, you can convince yourself that each one of those little dots doesn't live for very long. But if you do the same thing on the cell layer, you actually do see little like wavelets that live long enough to propagate their own distance, and kind of all line up in these streams and stuff. So I think short wavelength fluctuations do propagate like waves, and all of the other fluctuations propagate, or rather don't propagate. They're all overdamped, and sometimes it just sits still. So finally, we want to figure out their relationships. So I just thought, well, let's just go back to a correlation function. Let's say I can take those Fourier filtered movies and say, given a long wavelength fluctuation in the substrate, how long till I see a short wavelength fluctuation in the cell layer, or vice versa, or basically there's 4 combinations. So you can do that, and I think there is a logical sequence that has to happen. You pick one and you just walk from one to the next, and it's if you want to answer the question, how do long wavelength deformations end up translating into long wavelength migrations via some local fluctuations? It follows a logical order based on how these correlation functions evolve. Long wavelength deformations excite short wavelength cell fluctuations. Short wavelength cell fluctuations excite short wavelength deformation fluctuations. Somehow, I don't know how, short wavelength deformation fluctuations give rise to long wavelength migrations. This is the part I'm totally fuzzy on. I don't know how to think of it. I'd rather have a model. I'm trying to do this in a model-free way, but I'd rather have a model to understand this. So if anybody is marginally interested or has an idea, please contact me and we'll write a paper. I'd love to. So thank you very much.
Anybody wants to ask a brief question?
So in the first part of your talk, you showed the dependence of the correlation length on the density of the cells, and I guess you did it by just waiting while cells are dividing. Is that correct? On the substrate? Or?
Yeah. Throughout, so this is one really, really long experiment, and they're constantly dividing. But I chopped the data up into short enough data sets that I'm not dominated by.
My question was, how can you be sure that that is due to the density change and not maybe some extracellular matrix developing or cells are aging?
Yeah. No, that's a good point. I don't know. Our collaborators who did similar long time experiments like this stained for like fibronectin and things like and didn't see any kind of like large scale ordering or deposition. But I don't know. I mean that's totally possible. However, I'd have to say that it has to depend on the substrate because you see the opposite behavior on glass.
I mean, you can probably fix the cells so they don't divide, but you can squish them and
or you just prepare a new sample each time.
Any more questions? Well, let's thank the speaker again. Next, we have a talk by Thorsten Wellert from Syracuse University, speaking on F-actin dynamics in human skin cells and directing the velocity of cell migration. Anybody a Mac expert in getting this to display? No.
You're getting an entire corporation. No, I think okay. You have enough guys who are anti-parallel
Christina, there's a screen behind you. You have to click it out.
The problem is that, I don't know, it's very hard to
Especially when you want to simultaneously get small stuff.
Now we have two.
Can everybody hear me? Probably, right? Okay, good. First, I would like to thank the organizers, Christina and Roy, for giving us the possibility to present our data. And I would like to talk about a more or less biological problem about F-acting dynamics that directs velocity of cell migration. Because Margaret gave a really nice introduction yesterday, I just want to talk briefly about the cell migration itself. So this process is required for embryogenesis, tissue repair and wound healing, as well as immune responses. But it is also responsible for tumor formation and metastasis, as well as for some brain defects. Cell migration is a four-step process that includes the protrusion that usually occurs at the leading edge of a migrating cell, and the formation of new adhesions that occur also at the leading edge, and then the contraction of cell movement or movement of the cell body, and finally the dissolution or de-adhesion that occurs at the rear of the back of the cell. So this is an example of migrating keratinocytes, and you can really see that it forms protrusion at the leading edge, whereas it's retracting at the back. Here you can see actually the retraction fiber. This is, well, it's not so good to see probably. This is in a large region of a cell that was transiently transfected with cherry actin, and you can see actually besides membrane ruffling, the protrusion as well as the retraction. Besides actin polymerization that is required for cell migration, Rho GTPases play also an essential role in this, during this process, and there are primarily three major GTPases, CDC42, Rho A, and RAG1, that are involved in the formation of specific actin-containing structures. So CDC42 is responsible for the formation of filopodia, whereas Rho A is responsible for formation of stress fibers and focal adhesions, and RAG1 is involved in the formation of lamellar protein. All these Rho GTPases cycle between an active or a GTP-bound state where it can bind to effectors, or they can stay in inactive or GTP-bound state. For our experiments, we used the human oral keratinocytes. This is an immortalized cell line, and the first, or the image on top shows you an immunostain for actin in red and tubulin in green, and you can see that this is really a polarized cell with a leading edge where you have filopodia and membrane ruffles present, and at the back you have the action retraction fiber. At the bottom there are you can see a migrating cell visualized with DIC microscopy. Here's a little set of two dividing cells visualized by video microscopy. To study cell migration, today there are actually three ways, three common ways. The first and the classical one is the cell culture, where you have a cell culture vessel, grow your cells, and then monitor the process of cell migration for a certain time period. Then you have that wound closure or stretch assays that you can use to study cell migration. For this, you're using usually a tip, a pipette tip, produce a gap in the middle of a subculture dish and follow the gap closure over time. And then, I never used this actually, but obviously there were some labs that use fluorescent beads, and they followed these beads by fluorescence microscopy, and they were able to track those cells. The next slide shows actually one of these classical scratch assays that we have performed, and here you can see how it's working. So this is for a period of 12 hours, and I will pray again. So here you have the gap, and you can see that the cells or single cells start to migrate into this gap, and over time that this gap is really closed up with cells that are moving into this gap.
How big is that gap?
It's about 200 nanometer, I think. Oh, I'm sorry, yes, yeah. I'm sorry. Good. Because scratch assays are not the best solution in our opinion, or it's not really comfortable to use this, we decided to perform sort of long-term live cell imaging to study cell migration. And luckily, the microscope manufacturers made some toys available. So here we have tested actually both systems. So one is from Nikon, and this is a so-called biostation. And the other system was designed by Olympus, and is called the incubator microscope. I just want to talk briefly, or mention briefly, that this system is or contains phase contrast optics, and there's space for only a single 35 millimeter dish, which is sort of limiting, whereas the incubator microscope from Olympus provides a rotating disc inside the incubator, and there is space for up to 8 35 millimeter dishes, which allows you to do a lot of experiments simultaneously. And in addition, this system contains DLC optics, which allows you better imaging of the cell migration itself. So the next step is, when we've collected the data with one of these systems, then we transferred them into a Metamorph for further motion analysis. So what we have used is that track objects tool, which allows you to place rectangular regions in the area of interest, and over time you can follow the tracking process. So this would be here at the beginning, and this is where the tracking process is right now. And finally, you're ending up with a whole bunch of data that allows you to characterize the movement of individual cells or cell groups. So this, for instance, you can get data about position, velocity, as well as distances. In addition, we performed kymographic analysis to get further information about leading edge activities. So here we have a DIC image of a keratinocyte. We placed a couple of lines in the regions of interest, and you can create those kymographs or time distance plots, which allows you to follow the movement of the leading edge, and you can clearly distinguish between protrusion and retraction activity, as well as you can see that the structures that are moving. So in this case here we have a lamellipodium that is protruding and retracting, and here you can see a filipodium that is actually protruding or retracting. With this in mind, we have created sort of motility cell migration assay where we can monitor cell groups, not only single cells, cell groups, and this is a movie that covers actually 30 minutes, a period of 30 minutes, and these are, let's say, control cells that were cultured in 35 millimeter dishes, and you can see that you have protrusion and retraction activity, that the cells are sort of connected and circling randomly around. When you analyze this movement with the Metamorph software, you're able to create this migration path, and you can really see that these cells have a non-synchronous or randomly movement, and if you do this Kymographic analysis, you can see that the protrusion velocity or the instantaneous velocity ranges from about 1 to 1.5 microns. So this is what is pretty straightforward. So you can collect not only data from single cells, but only from cell groups. And the next step, we were asking, so how can we alter the cell migration? What can we do to get further information about changes? So and this is when we got in touch with microbiologists. And ladies and gentlemen, I can present now the second key player in our migration assay. And this is Candida albicans that we are adding actually to our cultures. Candida albicans is, as stated here, a human pathogen, and to make it straight, it can actually kill individuals under special circumstances. This pathogenic fungus exists in 3 isoforms. So typical, you have, it stays in the yeast form, like Saccharomyces cerevisiae. It can stay in an intermediate state and produce this so-called pseudohyphae, or really true hyphae, which represent a multi-nucleate filament. So when we added those fungus to our cultures, you can see that the migration of those human or keratinocytes is changing dramatically. The first thing I want to point out is that you can clearly see that the fungus is growing, so it forms really nice hyphae, and that the cell migration, or the cell sheet is migrating more synchronous, more directed in one way. We don't know whether this is chemotaxis or not, but obviously there's something going on with these cell cultures. When we analyzed these cells further, you can see indeed that these cells move as a sheet, synchronous, and it's more directed for some reason. So analyzing or using kymographs for further analysis, we were able to see that the instantaneous velocity increased dramatically. So now we're reaching up to 4 microns per minute for individual cells. Next, we plotted actually, or we created a graph where we plotted these instantaneous velocities and compared instantaneous velocities of untreated cultures versus cultures that were treated with C. albicans for 3 and 6 hours. And we were able to see that the untreated cultures and even the 3-hour instantaneous velocities for the 3-hour time interval showed a normal distribution, whereas at the 6-hour time interval we were able to pick up a bimodal distribution with actually 2 major peaks, one at around 1.5 micron, and the second one is around 2.5 micron. When we compared the migration rate of these cells that were moving under different conditions, so we can show, or we could show, that untreated cultures move at an average of 1 micron per minute, whereas cultures that were treated with C. albicans for 6 hours, they showed an increase of about 2. What's the time?
Another 3 minutes.
So the next thing we did, we looked more closely to the actin filament system itself, because obviously there are some changes going on that are triggered by the presence of this fungus. So we fixed and stayed cells with rhodamine phalloidine at defined time intervals. So when you look at the untreated cultures, you primarily, or you can see that those cells exhibited primarily philopodia. At 3-hour time interval, we were able to see primarily stress fibers and focal adhesions. And at the later time interval, these cells exhibited lamellipodia, extensive lamellipodia. In control experiments, we used Saccharomyces cerevisiae that were co-cultured with these oral keratinocytes. And as you can see, there's not a big difference between untreated cultures and those that were treated with Saccharomyces. And finally, we were interested in whether the actin dynamics is really affected. So we've transiently transfected these cell lines with YFP actin, and in this image you can see a single cell in an untreated cultures, and I think it's really nicely that the protrusion and retraction activity of philopodia visible, whereas in the presence of Candida albicans, you can see clearly that actin stress fibers and focal adhesions are present, and that the protrusion and retraction cycle is affected as well. So when we co-cultured these cells with Candida for 6 hours, you can see a really nice lamellipodium and I would say increased protrusion and retraction activity. It's actually the same play rate, so these are 10 frames per second. So, and finally, this is our control again, the culture that was treated with Saccharomyces cerevisiae, and you can see that these cells behave similar in terms of actin dynamics as untreated cultures. So, from this one we can conclude that C. albicans, pathogenic fungus, can alter the actin filament dynamics and can increase the cell migration of human oral keratinocytes at early time intervals. We did not observe actin remodeling when Saccharomyces cerevisiae was added to cultures of human keratinocytes, and we can make the statement that C. albicans probably activate the Rho-ROCK signaling pathway. Finally, I would like to show a working model. We have more evidence, but time is not enough. This diagram shows the sort of the sequential changes in the actin cytoskeleton caused by C. albicans. So it is well known, and we have shown that this fungus can secrete virulence factors into the medium and cause actually changes in the actin cytoskeleton. So we are starting with philopodia and microspikes in untreated cultures, but at later time intervals, we see that cells exhibited focal adhesions and stress fibers, as well as lamellipodia and membrane ruffles at the 6-hour time. So finally, I would like to thank some of our collaborators. This is work done by Christiane Rollenhagen, a postdoc in Paula Sundström's lab at Dartmouth Medical School. And this is our former technician, Mary Young. She has developed she made it possible to transfect those cells, since it's a little tricky because you have to keep them at a really low confluent level, and you can't use electroporation in one of these classical stuff that is available. Thank you.
Do you see the same results if you use fungus conditioned media and just add that without the actual pathogens?
We see this effect. It's a little bit delayed. So obviously, you need the presence of the fungus as well. So and I can tell you that these fungus or the fungi secreting SAPs, these are aspartyl proteases that are responsible for these changes. But it is not known what the receptor is on the cell. So someone can figure that out.
questions? Well, let's thank the speaker again.